Hello everyone and welcome to lecture 8. We're going to talk about how we move. We're going to talk about muscle contraction in the first part and how acetylcholine kicks that off. We'll talk about central pattern generators in the spinal cord, and then we'll talk about some higher level regulators of movement in the cortex and the cerebellum as well. Okay, first we're going to talk about muscle contraction. We're going to start in the muscles and we're going to work our way back to the brain. Hopefully we recall that acetylcholine is the neurotransmitter released by our motor neurons to cause muscle contraction. Okay, first let's go over the basic units of muscles. The basic building blocks or the individual units in muscles would be sarcomeres. Here we're looking at two sarcomeres, one that's relaxed and one that's contracted. What you're going to notice is that there are two types of filaments. There are the thin actin filaments and the thick myosin filaments. The thin actin filaments of one sarcomere will be joined to another with this sort of zigzagged structure called the Z-band. The myosin filaments are all contained within one sarcomere, and they're going to be held between the two Z-bands. When the muscle contracts, the sarcomeres shorten, and this is done by the thick myosin bands pulling on the actin bands. So in this illustration, the actin bands are shown in red, and you can see here that the two Z lines, or the two Z bands, are going to be brought inward toward the middle during contraction. And that's all that happens. There's a shortening of sarcomeres. Okay, and these are the building blocks of our muscles. Okay, so here's some sarcomeres again, now shown somewhat closer to how they look in life. Each of these sarcomeres is just a small component of a single muscle cell, the single myofibril. And by shortening these, we can generate our muscle tension. All right, here's a brief movie on how this works. So it's all about interactions between actin and myosin. Okay, that's the cross-bridge cycle here. There's our sarcomeres, and there they are shortening. When all the sarcomeres shorten, so does the entire muscle. Here we can see our thick and thin filaments. The thick myosin filaments are shown in the center, and the thin yellow actin filaments are shown on the outside. The myosin filaments actually contain little motor proteins, myosin, that pulls on actin, just like that. Alright, so these are just protein-protein interactions. It's all about the myosin-actin cycle here, the myosin power stroke. Okay, so that myosin filament contains a whole bunch of these myosin heads, and these are the motor proteins that move along actin filaments. They have two regions. They have the S1 region, that's this blue oval that we see in the cartoon. This is the part that actually does the work. The S2 region is this flexible connector that holds the myosin motors onto that whole myosin filament. So that'd be the thin black line here. In the myosin power stroke, we're going to be moving actin about 10 nanometers at a time every time we spend one molecule of ATP. It all comes down to ATP. This is why we need our glucose. So we can make ATP. Not only do we spend it in neurons to keep the sodium, potassium, ATPase going, but our muscle cells are also going to use a ton of ATP to allow our muscles to contract. All right, let's walk through this. Okay, we'll start off at location 1 in the bottom left. We have our myosin bound with ATP. The myosin head is an ATPase. It can hydrolyze ATP to form ADP in one of those phosphates. So instead of adenosine triphosphate, we now have adenosine diphosphate and an additional phosphate that was broken off. When ATP is hydrolyzed like that, so when we move from 1 to 2, okay, from bottom left to upper left, you can see that the orientation of the S1 region of myosin changes. It's now no longer cocked back. Now it's shifted forward. Myosin ADP can bind to actin, and when it does so, this interaction is going to destabilize the interaction between ADP and myosin, as well as the phosphate and myosin. So that interaction that we see, the binding of myosin to that actin filament, shown at the top there, that's what AF means, actin filament. So as we move from 2 to 3, upper left to upper right, we have the interaction between myosin and actin. That, as we can see, is going to cause the release of ADP and that phosphate as we go from 3 to 4, so from upper right to bottom right. That's the power stroke. When we release ADP, that causes actin to shift, that causes myosin to shift its orientation, pulling on actin. Since we no longer have ADP bound, we can rebind a fresh ATP. That's going to break the interaction between actin and myosin. ATP gets hydrolyzed, myosin pushes forward. It binds to actin, releases ADP, kicks backward. That's it. That's our myosin power stroke. And through many repetitions of this, that's how we go from relaxed to contracted. All right, here it is in cartoon format. We have our myosin head on the bottom, actin filaments on the top. So we're going to bind, we're going to form that cross bridge. That's all that is. That's the interaction between myosin and actin. All right, so now we're going to release our ADP and phosphate. All right, there went the phosphate. We bind, we release our ADP. Once we release that, then the myosin head kicks back. All right, this myosin head doesn't have any adenosine nucleotide attached to it, so now it can bind a fresh ATP. There's our ATP binding to myosin. When ATP binds, that destabilizes the interaction between myosin and actin, and it unbinds. Remember, myosin is an ATPase, so it hydrolyzes ATP to form ADP and a phosphate. In doing so, that's going to cause the shape of myosin to change. So now it's going to cock forward. Myosin can interact with actin more stably than it can with ADP, so it's going to rebind and it's going to continue over and over again. So if myosin and actin are able to interact like this, why aren't our muscles always contracted? The simple answer is that they can't always bind to one another. There's a little bit of logic worked in here. So that the binding site for myosin on actin is normally covered. It's normally covered by tropomyosin. So when we're resting, the myosin binding site on actin is going to be occupied by this filamentous protein called tropomyosin. In order to move tropomyosin out of the way, we need to elevate intracellular calcium levels, which are normally very low. Once we have an increase in calcium, calcium is going to bind to this other protein called troponin, which pulls tropomyosin out of the way. Muscle cells are filled with calcium in this sort of modified endoplasmic reticulum called the sarcoplasmic reticulum. Once we have release of calcium from there, calcium binds to troponin, pulls tropomyosin out of the way, and the myosin binding site is exposed. Once the myosin binding site is exposed, we can have our myosin power stroke. All right, so here we can see the activity of calcium. So there's our calcium ions coming in. Once we have an increase in intracellular calcium, calcium binds to troponin. That's going to then pull tropomyosin out of the way. Once it's bound to calcium, remember calcium has a charge. That charge is going to change the shape of troponin. It's going to pull tropomyosin out of the way, and now we have our actin binding sites exposed. We can have our power stroke. Where does the calcium come from? Hopefully we've all guessed that it's going to come from the release of acetylcholine. So motor neurons are going to stimulate calcium release within muscle cells by releasing acetylcholine. This is how our motor neurons can control our muscles. They release acetylcholine, it binds to nicotinic acetylcholine receptors, and we get an increase in intracellular calcium. All right, and that's what we're seeing here in these data. These data are showing us an increase in intracellular calcium as a result of synaptic input. So the black line is showing you the postsynaptic currents. The red line in panel A is showing you the membrane potential. So we can see a number of action potentials occurring. B is showing us the relationship between calcium, the postsynaptic currents, and the cell length. Okay, so muscle cells can fire action potentials themselves. That's what we're seeing up at the top. And when they fire that action potential, that depolarization is going to cause an increase in intracellular calcium. We can see an increase in intracellular calcium in panel B. The blue trace is showing you an increase in fluorescence for a calcium indicator. So when we get the calcium spike, look what happens in the trace below it, the cell length. The muscle cell shortens. The cell contracts whenever we have an increase in intracellular calcium. The more calcium we have, the greater the contraction. All right, so let's see how this works in cartoon format. We have our motor neuron shown on top. It's not active at this point. We're not releasing any acetylcholine. All right, our muscle cells are nice and long. When the motor neuron fires, it releases acetylcholine. Acetylcholine binds to nicotinic acetylcholine receptors. They have some degree of calcium permeability. They're nonspecific cation channels. That depolarization that occurs is also going to activate voltage-gated calcium channels on the muscle cell. This calcium is going to cause that calcium-induced calcium release that I don't think we covered in this class actually. So the calcium is going to bind to ryanodine receptors and cause the further release of calcium from the sarcoplasmic reticulum. The end result is a substantial increase in calcium levels within the cell. That moves tropomyosin out of the way when calcium binds to troponin, and that allows the shortening of sarcomeres by the myosin power stroke. This is a very straightforward process, but this is how our motor neurons control our muscles. It's just that simple. In order to allow reliable control of the muscles, the synapses that form between motor axons and muscle cells are huge. These are much, much larger than the synapses you're going to find in our central nervous system, which are very small and very weak. The neuromuscular junction is actually very, very large, and it has some specializations that allow acetylcholine levels to remain elevated. So first of all, there's about 1,000 active sites, so we're going to have many, many sites where we release neurotransmitter at the presynaptic site. So a single action potential is going to cause the activation of hundreds of different active sites and the release then of hundreds and hundreds of acetylcholine-containing neurotransmitter vesicles. The postsynaptic site, as we can see in panel B here, has these postjunctional folds, these sort of invaginations of the postsynaptic membrane that are designed there to keep acetylcholine from diffusing away from the synapse. So if you look on the bottom, you'll see those infoldings, especially on the left-hand side. So the postsynaptic cells shown on the bottom. Of course, we can tell the presynaptic cells on top by the presence of neurotransmitter vesicles. The postsynaptic cell, you can see, has those infoldings, and that's going to trap acetylcholine. Not only do they have a high density of nicotinic acetylcholine receptors, they have these postjunctional folds that trap acetylcholine. So when we have the release of acetylcholine, we release a substantial amount because of that large number of active sites. That acetylcholine gets held in the postjunctional folds so they can activate the high density of nicotinic acetylcholine receptors. So we get reliable activation of our muscles. All right, so that's how our muscles work. Let's move back a bit though and talk about what's going on in the spinal cord to allow the activation of the correct muscles. Because our spinal cord actually has some processing power. This is a part of the central nervous system. Okay, it's not just a simple input-output. There are what we call central pattern generators that are there to help create stereotype motor movements. So those central pattern generators are just a collection of interneurons that will create a given pattern of motor activity based on the input that they're receiving from sensory neurons, or the input they're receiving from the higher areas of the central nervous system in the brain. So these central pattern generators are going to take the input from upon high, from the cortex and others, and the input from sensory neurons, and use that to help control our muscles. So the simplest central pattern generators are going to just carry out the simple reflexes that we've already learned about. So the stretch reflex has a very simple central pattern generator, just a single inhibitory interneuron there. So whenever we have stretch of a of a muscle, this reflex is going to cause contraction of that muscle to help maintain a relatively stable muscle length, unless we decide to change our muscle length and move our bodies. But any erroneous stretch of the muscle is taken care of by this reflex. So we should keep in mind that we have two different types of muscle fibers. We have our extrafusal and intrafusal. Here we're just talking about the movement of our extrafusal fibers. But the intrafusal muscle fibers are also very important because they're going to be innervated by the sensory neurons. So this is where we sense our stretch. So in that patellar reflex, when you get a tap on the patellar tendon, that stretches the muscle fibers, both extrafusal and intrafusal. Now when that muscle stretches, the sensory neurons get activated in response to the stretch, and they're going to release glutamate onto, you can see the extensor motor neuron, as well as that blue inhibitory interneuron. It's going to release glycine. So let's follow the simple route here. The sensory neuron activates the extensor motor neuron that then activates the extrafusal muscle fibers on the extensor muscle and causes us to kick our leg. To make this a smooth response, that central pattern generator is going to translate the excitatory glutamatergic input it receives into inhibitory glycinergic output onto that flexor motor neuron. So the flexor motor neuron gets inhibited, and that prevents activity of the flexor muscle from getting in the way of the shortening of the extensor muscle. And that's all that this central pattern generator does. Carries out a simple reflex. We have other reflexes that are mediated by our tendons. So there's this Golgi tendon organ found within the connective tissue that holds our muscles onto the bones, the tendons. The stretch receptors there are also hooked up to central pattern generators. Just a very simple inhibitory interneuron. It's the exact same thing. All right, this is the exact same reflex, slightly different function. The muscle stretch is there to make sure that our muscles aren't shortening or lengthening without us telling them to. So if we miss a step that we think is there, the muscle is going to continue to change its length, even though it shouldn't. We'll pick that error up and fix it. The Golgi tendon reflex here is to prevent damage to our skeletal system or other soft tissues. If the muscle is contracting so hard that we're starting to stretch on our tendons, we should probably tone that down, and that's what this reflex does. So whenever we have stretch in the Golgi tendon, we're going to have activation of sensory neurons, and here we're going to inhibit the motor neuron that would cause contraction in the in this area, and thus stretch on the tendon. So if we're shortening that muscle that we see here, and we start to pull on the tendon, activation of those stretch receptors recruits that inhibitory interneuron shown in the cartoon that's going to help turn down the activity of that motor neuron so that it stops causing muscular contraction, right? Because we're potentially going to injure ourselves because we're now starting to stretch our tendons. So the muscle is just putting out more force than we might be able to handle. So this reflex prevents us from damaging that connective tissue. And then, of course, there are more elaborate central pattern generators, and that's what we can see here. So they're showing you some of the upper circuitry, but then in those little boxes near the bottom, those are the central pattern generators. So these can also be used for more complicated movements. Usually these are involved with rhythmic motor movements, like walking, for example, or if you're a lamprey from swimming, so moving your muscles in an alternating fashion so that you swim effectively. How do we do that? Well, it's with this central pattern generator. All right, now let's see how this central pattern generator works. What you'll see here is E is excitatory, M is motor neuron, and I is inhibitory. So there's these inhibitory connections between our two central pattern generators, and both of them are controlling the motor neurons. Okay, let's go over what they're doing here in a little bit of detail. So we're going to coordinate our movements with both excitatory and inhibitory movements. We're going to try to create alternating patterns of activity. Okay, so we have our big yellow excitatory connections. Those are the excitatory neurons. And then we have our little blue inhibitory neurons. Okay, so once the excitatory neurons become active, they continually drive the activity of their neighbors. So we don't just get one activity. Here we get repetitive activation of these excitatory neurons. That repetitive activation is going to increase extracellular glutamate levels far more than just one or two action potentials. So the nearby motor neurons are going to fire. Okay, so that's what the repetitive activity is doing. This is also going to drive the activation of that nearby inhibitory neuron to keep the other excitatory neurons from being active. So here, when one side of the body is active, the other is inhibited. And that's what's going to allow the lamprey to swim, to contract one side and relax the other, then contract that opposite side and relax the other. Or when we walk, we're going to be moving one leg and then the other. Okay, and a lot of walking is actually carried out far below your brain. So a lot of it's going on in the spinal cord. Now, you might think that once those excitatory neurons get going, well, they should never stop, right? And of course, that's not the case. As we can see from this cartoon, they eventually stop. So they drive this excitatory burst, then they stop. Then they drive an excitatory burst, and then they stop. Part of that has to do with inhibitory input, but part of it also has to do with them turning themselves off. So the excitatory burst firing has to be halted. And the way that that occurs is just through some intrinsic properties of the neurons themselves. So they have calcium-gated potassium channels. So once they start burst firing, well, that's going to activate voltage-gated calcium channels. They're going to get an increase in intracellular calcium levels, and that is going to increase their potassium conductance, which then leads to shunting inhibition and turns off that burst firing. All right, and we can see the data here. In panel A, we're looking at a normal burst that turns itself off. And below that, they're plotting the intracellular calcium levels that they recorded with a calcium-sensitive dye. In panel B, they've applied the voltage-gated calcium channel inhibitor is radipine. After they fire the burst, you can see there's not an increase in intracellular calcium levels, and the neuron remains highly active after the burst as opposed to the top. Right, so this neuron is much more active because it can't have that increase in calcium that drives the increase in potassium conductance. So the excitatory neurons are there to drive burst firing and then turn themselves off. The inhibitory neurons are there to create that alternating pattern between the central pattern generators. So you can do left, then right, then left, then right. Because as those excitatory neurons are burst firing, they're driving the activity of that little blue inhibitory neuron, and that keeps the excitatory neurons off in the corresponding central pattern generator on the other side of the body. So the excitatory cells themselves are driving this, the burst and then the pause. And the inhibitory neurons are helping to coordinate this between the two sides of the body. This is what's going to allow central pattern generators to coordinate some fairly complex movements without the input from the brain. All right, we can see this here. So central pattern generators in our brainstem and spinal cord are going to allow us to carry out these simple reflexive behaviors like breathing and swallowing or even walking. So on the bottom here, we can see a decerebrated cat. In other words, a cat that doesn't have its cerebrum. So everything from the brainstem up is gone. What they've done is sever the connections. So now the spinal cord and brainstem are intact, the cerebrum is intact, but the cerebrum can't communicate with the spinal cord. So this cat is actually paralyzed, but what they've done is support it with a little harness to support its body weight, and they're going to run a treadmill underneath it. All right, you can see the treadmill moving now, I hope, and the cat's taking steps.